Hello, everybody. My name is Divya. I'm a biology faculty at Azim Premji University. Today, we are going to be looking at cells. So, cells are really important, and we are going to be looking at various aspects of cells. Why are they important? Why are they kind of small in size, and how big can they get? Is there some kind of a limitation for how big they can get? And what are the various functions that a cell does? And how does things move from one part of the cell to another part of the cell? So, we'll also learn about various cell organelles inside the cell. But before going into these nitty gritty details, I want to take a minute to tell you about a story. This is a real incident. So there was a 25-year-old girl who was very, very athletic. She was running regularly. She was very active. She participated in a lot of stuff and she was very energetic. And then when she turned 25, she realized that she was suddenly becoming very tired. She could not even, in fact, walk a few meters. She would feel very tired, even if she walked a few meters. So then she went to a doctor, and later she found out that she has some kind of a mitochondrial disease. So this kind of illustrates that even small organelles such as mitochondria is extremely important for large scale effects, large scale functions. And this kind of shows why cells are really important. So there are several diseases which are defects of various cellular organelles. Of course, we won't go into that, but that kind of illustrates why cells are really important, because cells are basically building blocks of all the organisms. So all of us, all living beings are composed of one or more cells. They can be unicellular, where there is single cell performing all the activities, or it can be multicellular organisms where multiple cells get together to form an organism. So cell basically is the basic unit of all living things. And it's really nice to see that cell kind of compartmentalizes life from the external surrounding. So whatever happens in the external surroundings, it doesn't affect the cell immediately. Of course, there are various adaptations that the cell would have had in order to kind of live in its external environment. Cells also arise from pre-existing cells. That means that one cell will give rise to another cell through reproduction, right? So these are certain basic facts of cell theory. And cell was discovered in the 1600s by Hook, where he was looking at a cork. So this is a plant material under a microscope, and he saw very, very structured blocks, and he called them cells. And from there onwards, the journey of finding out what are the subcellular components and other things started. So let's look at how big these cells are. So cells are microscopic. Most cells are microscopic. So say, for example, a plant or an animal cell, if you look at it, those are around 10 microns big. Bacterial cell is even smaller, which is around 1 micron large, whereas frog eggs are slightly bigger, or even human egg, in comparison to your regular plant or animal cell. This is a nice representation because it kind of put things in perspective. So we are about 1 meter. So if you take a average height of a human being, that's in meters, right? And a ship would be 100-fold, so around 100 meters long. So when compared to that, cells are really, really, really tiny. So this is a relative size chart based on a logarithmic scale. Okay, so now we know that cells are rather small. But what is the reason? Why can't cells grow bigger than that? Why can't cells be as big as this room or as big as a house, for example? There are certain limitations. So think about that. So a cell, basically, I told you earlier that it's a contained unit, right? It separates whatever is happening inside from the external world. But there are a lot of things going in and out of a cell. So there are molecules entering cells, and then there, its products are created, and that has to be, put out of the cell, right? So there are a lot of metabolism which is happening in the cell. There are movement of particles in and out of cells. So all those things are limited by the surface area of the cell membrane. So there's only so much, so many, particles that can enter, because the surface area is of one size or a certain, range. But if, say, for example, if in order to kind of increase the surface area, certain cells, certain specialized cell can have very, very convoluted or very ridged surface, so that increases surface area, thereby allowing a larger amount of molecules or particles to enter in and out of the cells. So those are some of the adaptations that some of the cells undergo in order to increase the surface area. But basically, cells are limited by surface area. One more reason is we kind of think about cells as two-dimensional structures, because that's how our textbooks are depict them, right? We kind of draw them as like these round, and we put in certain organelles inside it. But cells in reality are three-dimensional, right? It has volume. It's not just one flat thing. It's a three-dimensional object. So volume of a cell, so if you think about cell as a sphere, volume increases much more rapidly. Volume of a sphere is much more than the surface area. So as the cell grows, the volume increases much more rapidly than its area. So that also limits the cell size. It can't grow really big, so that limits the cell size. But certain cells are really long. For example, nerve cells of giraffe is one cell can be really long. So in comparison, I've put a basketball field here, which are 10 feet. So certain neurons can, in say larger animals like giraffes, can be very, very long, but of course they're super thin. So that's just one example of certain exceptions. Okay, now let's look at certain shapes of cells. I have written here that shape of the cell reflects its function. So here are three examples. Red blood cells are kind of flat, and it doesn't even have a nucleus, because it is utilizing all its area, all its whatever is present. The cell entirety is filled up with hemoglobin, and also to carry oxygen, right? So it lacks nucleus, and it's kind of flattened, and nucleus is bulky, so it helps to get rid of the nucleus, and it's flattened. That helps it to go through very, very narrow blood vessels in order to carry oxygen and give it to different parts of the body. So that you can see that structure reflects its function. Nerve cells are another example. They're very thin but elongated. So in order to, conduct electrical impulses. And pollen cells, very, very interesting cells. It has bizarre, very beautiful structures. And these pollen grains have basically kind of spiny, these kinds of structures, because one of the reason is because it increases addition to the stigma, the female reproductive part in the plant. So if you want to stick the male reproductive part to the female of a plant, then it makes sense for pollen grains or pollen cells to be very sticky, so that it can have different these kinds of shapes, so that it can stick to the stigma. Okay, so that's about different kinds of cells, and also cells have different shapes, right? So we are going to look at more closely into what happens in the cell and what is the function of a cell, right? So cells are like tiny factories, and the key here is they kind of perform different organelles perform different functions. So division of labor is the key to be so efficient. So cells are really, really efficient. So let's look at some of the, the factory-like work that the cell does. So you have cell membrane, which covers the outside, that acts like a gatekeeper. So cell membrane controls what goes in and what goes out of the cell. So that acts as a gatekeeper. Nucleus is the most important, one of the most important structures in the cell. So that is like an operation center. A lot of decisions are made in nucleus, which contains DNA, of course, and that's like a instruction manual. Okay, if you want to produce certain material, these are the instructions to be followed, and that's what DNA does. Mitochondria are the powerhouse of a cell, of course. So you want to produce certain things, so the energy is generated by mitochondria. Golgi complex, on the other hand, is a packaging unit. Okay, you have produced something, it has to be packed and it has to be delivered to certain area. So Golgi packs the protein, and wherever it has to go, it will send that to that particular region. So it's a packaging unit. Endoplasmic reticulum, on the other hand, it's the manufacturing unit of the cargo. So I'm saying this as cargo. So what I mean by cargo is various kinds of proteins and sugars and other things which are actually produced inside the cell. So the manufacturing unit basically happens in endoplasmic reticulum. Say suppose some cargo or protein needs to be degraded. That happens in lysosomes. And how does these things move from one part of the cell to another part of the cell or beyond, outside the cell? This are facilitated by cytoskeleton tracks. So in the cytoplasm, there are cytoskeleton laid out. We'll talk about this later in this talk, where on the tracks, these cargoes are, it facilitates movement of the larger molecules. Okay, so these are various functions of the cell. Of course, there are several other components inside the cell which I have not actually talked about, but you can easily look them up in your textbook. So now let's look at, relative volume of, various of these major intracellular components. How much of volume, percentage of total cell volume which is occupied by these intracellular components. This is with respect to a liver cell. So a large amount of it, 50%, or slightly greater than 50%, is cytosol. That's just the cell soup, right? And about half of it is around 20%, 22% is also occupied by mitochondria. And you can see that ER and Golgi are all relatively small amounts, right? And even nucleus occupies a small amount. So you might think that, cell is largely kind of watery and, most of it is empty, but that's not the case. We'll talk about how packed cells are in a bit. But the other point I wanted to make was that when we draw a cell, we're always used to drawing cells with like one big nucleus and one mitochondria and one Golgi complex and stuff like that. But remember that depending on the need of the cell or the type of cell, there are several mitochondria. And so it's not that there is only one mitochondria, there are several of them. And everything is connected to everything else. So all these intracellular components are connected to each other, and they are also extremely crowded. So this is this is just a graphic representation of an image showing macromolecular crowding. These are various kinds of macromolecules. There might be sugars, there are proteins, there are cytoskeletal tracts, where it shows how compact and how crowded cells are. And also within this, molecules are moving really, really fast. So to give you an example, inside of a cell is very crowded. So for example, an E. coli, Escherichia coli, cell has around 400 mg per ml of macromolecules. So that's really compact, and there's a lot of things inside a cell. And because of this crowding, there are chances of molecules interacting with another molecule is very high. So for example, here this molecule is sitting with the next molecule, so it's constantly colliding with the other molecules, and it's also undergoing random walk, and because of that there is diffusion, and that's how things are also moving, right? So and there are several small molecules, and they're moving very, very rapidly. For example, small molecules like glucose can move around 200 miles per hour, and larger molecules or larger proteins are moving at 20 miles per hour. So I'm talking about miles per hour. Of course, this is just to give a perspective of how fast cells, macromolecules might be moving within the cell, but of course it's not miles per hour inside the cell, but this is just to give you an example or put things in perspective. So of course it happens per second. It's very, very fast. The movement of molecules inside the cell is very fast. Okay, so now we are switching gears and talking about how does cargo, that is proteins here in this case, are transported from one region of the cell to another region of the cell. So I'm not going to be talking about protein synthesis here, but once it is synthesized in endoplasmic reticulum, how it moves. So these are the ways in which it moves. This is particularly with keeping in mind an excretory or a secretory protein, which kind of goes outside the cell. But similar principles apply even if the protein remains inside the cell. So first it is kind of synthesized with ribosomes and endoplasmic reticulum, and then that is kind of moved to Golgi, and from Golgi apparatus through several vesicles, it is transported to the plasma membrane, and then a protein is secreted outside. So let's look at some of the large dynamics which happens to move cargo from one place to another place. Again, I'm not going into details, but I'm just going to talk about certain things you may not have heard about in a traditional textbook or in your 12th grade. So there are when proteins are produced or synthesized on endoplasmic reticulum, there is basically movement of the cargo from endoplasmic reticulum to Golgi and from Golgi to back. So there is bidirectional traffic of material between membrane components. So this is endoplasmic reticulum and this is the Golgi complex. And you see that these vesicles bud off. These are vesicles. These are basically extension of these membranes. So all these organelles are membrane bound, right? So the membranes bud off of ER and they are moved to Golgi and they fuse or delivered to Golgi complex, right? And then after the function of these vesicles are over, then simultaneously these vesicles also bud off from Golgi and they come back, they are delivered back to ER, carrying with them trafficking proteins that are needed to be recycled for more rounds of transport. So new molecules are produced and they have to be transported from one organelle, intracellular organelle to another. So they are carried by some other kind of protein material and they're carried to Golgi, and then once that particular protein or cargo is out, then the proteins, the trafficking proteins are recycled back to endoplasmic reticulum. So what I'm saying here is there is continuous bidirectional traffic between different cellular compartments. What is really interesting is that some few years back, people had thought that Golgi apparatus can't be can't be disassembled. That means that Golgi is there in every cell. Golgi is just there. It can't be, destroyed, can't be created or destroyed, kind of a thing. But scientists have recently found that if you put in a drug called as Brifeldin A, it's a particular drug that makes Golgi complex kind of explode, and all the fibers, all the Golgi complex basically disappears within seconds, and the components, all the components of Golgi goes back to the ER. And of course, if you take away the drug, the new Golgi complex is formed, from the enzymes which are present in the ER that facilitate production of new Golgi complex. So when I was reading this, I was really fascinated by this information. So I just wanted to tell you about this, that Golgi, it's not that Golgi is always present, but Golgi complex can be blocked by a drug, and it can be completely disassembled. And of course, it can be also reassembled back. That's just an extra information. So now let's look at some of the other organelle. Now I'm talking about mitochondria and endoplasmic reticulum. So keep in mind, see, I'm talking about how various things move, right? So it's not like their organelles are static. We already talked about how, very crowded cells are and how things are moving really fast. Even organelles are exchanging materials and also talking to each other. So here is a section of the picture here, which is kind of highlighted here. So this is a part of, there is mitochondria, membrane of mitochondria, and also ER. So what happens is there are certain proteins known as tethering proteins. That means that they can kind of tie the ER and mitochondria together. Here, they bring the mitochondria and ER together to facilitate greater exchange of things like lipids or calcium and whatnot. So that's what is depicted here. These tethering proteins help the organelles to come close, and they exchange materials. So during glucose deprivation, so if the cell doesn't have enough glucose, mitochondria actually takes up lipids and calcium from ER, and in turn, that helps downstream events. So this is just an example that you should not think about cell as this static, fixed, kind of a unit where various organelles are present in it, and it's not like that. A lot of things are happening. It's a dynamic interplay between these organelles, which facilitate production and movement of molecules from one place to another place. Okay. Next is I wanted to tell you about how do these organelles move through inside the cell, and that is facilitated by microtubules. That those are one of the examples of cytoskeletal filaments. This is it's basically, this is a fluorescent microscopy image of a microtubule track. So imagine this is like a highway, or it has many roads, and there are certain highways also, and organelles move on these networks, on these tracks. So to give you a better understanding, these are microtubules, and there are proteins which are such as kinesin, which are moving along the microtubule. In fact, they're sprinting along the microtubule, and so you can see it almost looks like a step, and they're also carrying different kinds of cargo, or they're transporting membrane and different membrane bound vesicles. So this is how organelles move from one place to another place through cytoskeletal tracks. Okay, so this is a video about the Golgi disintegration. Please make sure to watch this video. It's fascinating how Golgi disappears completely and then reappears when the drug is removed. So to summarize, all living things are made up of cells. cells are really small. They are limited by the surface area to volume ratio. They are three-dimensional. They're not as 2D as we depict in our, in our textbooks. Cells function like miniature factories. They divide up work. Different cellular organelles do different things, and inside cells are really crowded, and molecules move very fast. And there are various functions for various, various organelles, and structure really determines function of a cell. Thank you.